Hello everyone, this is Malay. Today I'm just going to do a quick review over transcription and translation. So we're first going to work with transcription. Okay, so I'm going to draw a diagram of the nucleus. So this big circle is the nucleus. And I'm going to just highlight these regions. And what these are is our nuclear pore. Mind you, this is the nucleus. Now, inside the nucleus, what happens is something called transcription. So this is our DNA strand. I'm going to actually work super hard on this strand because this is going to be very important. Okay, now I'm going to make the corresponding for each of them. So we're just going to label these. This is going to be cytosine. This is going to be guanine. Yellow is going to be adenine. These are, and then green. What's the color? Red's going to be thymine. So we're just going to find the corresponding for each of them, right? So this would correspond with these. We're just looking for the complement right now, so don't worry about that, right? This would complement that. This would complement with that. Dark purple. That would complement that. That would complement with that. That, and then to end it all off. And then, okay, so this is our DNA molecule. Now, what's really important is I'm going to duplicate this. Duplicate. Now, oh, my bad. Now, what's going to happen is we're going to start splitting this apart. So, let's say we cut this in half. Right. So this is 1/2. Now, if you guys remember DNA replication, we have something called DNA polymerase, which would come in and make the complement of 1/2 of the DNA. Now, the same concept is going to be happening here, where we're going to have something instead of called DNA polymerase, we have something called RNA polymerase. I'm going to write that down real quick. And what RNA polymerase does is that it comes in to one of the strands. So imagine as if this is a DNA replication strand. So I'm going to just quickly draw this. Okay, you already know helicase is coming in, trying to break it down. Topo's in the front. You know the whole drill, right? But instead, let's say that they're just complement pairs. I'm not trying to make it all fancy and all. What happens is DNA polymerase comes in, and remember, primase also exists at this point. Primase signals RNA polymerase. My bad. I'm gonna confuse that a lot. So if I say DNA polymerase, just understand that means RNA polymerase. RNA polymerase comes in, right, and it makes the complement based off of RNA. And remember, RNA does not follow this setup exactly. Instead of thymine, it's uracil, right? So it's gonna make the complement of it. Right, and it's going to make the complement DNA. We're going to show red as a complement of the mRNA DNA. Okay, so they made the complement based off the mRNA from the DNA. So in this example, if there was a T somewhere, in this end it would be a U. An A. If it was an A, the complement would, instead of being a T, it would be a U. So they just make the complement of 1/2 of the DNA, and then that gets exported out. You might be and then the DNA goes back to normal. There's nothing so complex about this. Remember, all we need to understand and what we're going to need vitally from a source is that there's RNA polymerase. And what RNA polymerase does is that it creates a complement of the DNA strand and it's the complement in the RNA format. So instead of thymine, it's just uracil. It's only single-stranded, right? So what's happening is we're just gonna have this left. So let's say this is our RNA. These lines are nitrogenous bases. This is the sugar phosphate backbone. What happens is this is now unprocessed. Unprocessed basically means this RNA has not been turned into a protein. And what happens is it exits out of a nuclear pore. So I'm just going to write that down real quick. Exits from nuclear pore. After it exits the nuclear pore, we are left with the mRNA strand. Now, this is in the cytoplasm, by the way, so I'm just going to draw a highlight as if this is currently in the cytoplasm, right? Okay, so anything in this region is cytoplasm. Okay, and it finds something called a free ribosome. Now, if you don't know what ribosomes are, they're the centers in the cell which create proteins. Now, ribosomes are extremely tiny and they actually look like dots. It is like this. But if we were to zoom in on them, okay, they actually look something like this. With a larger subunit for L and a smaller subunit for S. Now, this is a setup of a ribosome. And it will find a free ribosome. A free ribosome is a ribosome that's not connected to any other organelle. If you remember a while back, we learned something called rough ER. And rough ER are studded with ribosomes. Now, mRNA does not go to that ribosomes. They go to the ribosomes that are not connected to anything, and they just freely wander around. For example, that's why they're called free ribosomes, free from any organelle connection. So they'll find that in the cytoplasm, and there's an abundant amount of them. And mRNA, which we're going to go to red, will actually find one and actually wedge in between the two subunits. Now, we're going to zoom in onto mRNA, the ribosome, and how they translate. Before we start, we need to know some basic understandings of how mRNA works. I'm going to draw a proper diagram of mRNA. Okay, this is going to be A, this is going to be U, and this is going to be G. Wait, no. I'm going to have to change this up a little bit, my bad. This is for the example, by the way, so understand. Okay, this is going to be A. Whoa. A, U, G, C, U, and then G again. Now, this is definitely not the full mRNA. It's much longer. But we need to understand a few vocabulary words. The first vocabulary word is codon. That's 3 nitrogenous bases. This basically means if there are 3 nitrogenous bases, for example, CUG and AUG, that translates into 1 codon. Okay, then we have something called start codon. A start codon is basically like primase. Remember how we talked about primase signals the DNA saying, hey, man, start here, start your DNA replication here. This is the same thing for translation. Remember, ribosomes are dumb. They don't know where to start for making the protein. If they start in the middle of the mRNA, there's vital portions of the protein that's not going to be synthesized. So what this does is the start codon tells them, hey, we start here. We start at this portion of the mRNA. Now, what is the start codon? It's actually a specific kind of specific kind of codon. So as I told you, codons are 3 nitrogenous bases. Now, there's a type of codon that translates to a type of protein. So, a type of amino acid. So, 1 codon equates to 1 protein. Now, there's a type of codon that doesn't equate to a protein, but instead starts this process of translation. And that's called AUG, aka this portion. This is considered as a start codon. Now, Ms. Suarez probably might ask this, so I just want to pull this up. RNA codon sheet. we need to understand how to read these. Now, mind you, she's not going to make us memorize this. That's just insanity. But it might be a question asked because we had worksheets on it. This is a horrible image. Okay, this is perfect. So these help us understand how the amino acids, how the codons are set up, and what the mRNA is translating. As I told you, AUG is a start codon, and AUG is right here. Now, MET also stands for start codon, and if we look here, same thing's happening here. It's a start codon. Okay, so there's specific codons for specific amino acid, and there's specific codon for the end of an for the end of mRNA, and those are called stop codons. Now, the stop codons are these, as you can tell, and there's also in here. Now, to read these is a little complex, but it's actually not that bad. So we're gonna first start with the boxing because I know how to do that a lot more better. All we need to know is see the codon, so CUG, for example, and then we find it here, right? So we see CUs, we see CUs, we see CUs, right? Because we see a C and a U, and then we see CUG, and that's connected to LU, which it doesn't look the best here, but that's a LU amino acid. Now, to read these, it's a little bit more complex, but it's actually not that bad. So we're gonna look at C, right? So we're trying to find LU. Right, so you can see how LU is right here, right? So we have to look in the sectors of the regions, so that we see. Oh, this is a horrible image. I'm so sorry. I just can't read it. I need something that I can easily read. No, is this better? Yeah, it's a lot better. All right. So to read these, we just look at where it starts. So we see that this portion is for all U's. So these are going to start with a U's. We're going to see this portion. Okay, so this 1/2, all of the amino acids are going to start with U something something. This portion is going to start with C something something. And same with A, same with G, okay? So with this specific case, we're looking at CUG. So we're looking at C specifically. Now we have to see the next number with it. So we see C, and the next number is U, right? So then we look at this portion here. Our next number are not G, it's not A, nor is it C, but it's U. And then this, okay, then goes to, we go here to here, and then we go here, and then we see G is here. And this basically means that our thing is a LU amino acid. So that's how you just read these charts. You don't need to memorize how to technically read them. You just need to know how to read them. You don't need to memorize every single amino acid and their combination. Anyways, now we're going to have to start seeing how this mRNA strand will create a protein. Let's go back to our ribosome. So inside our ribosome, we have something called tRNA. tRNA is the molecule or the structure that allows the translation from mRNA to protein. Now, tRNA, Miss Wars might draw it differently. I personally draw it like this. And then it has something called this. Now, what is this? Now, this part is really important. This is our thing. Now, this is a tRNA. I'm going to write that right here. And tRNA has a few main parts. So the most vital important part to recognize is this portion. This is called our anticodon. An anticodon is the complement of our provided codon. So for example, when we have this codon, AUG, our complement, let's see if you know, is going to be, instead of TUAC, right? That's our complement for AUG, right? And then that complement is considered as our anticodon. And that's going to be what this is here. Now, for one tRNA, it equals one anticodon. And it will only correspond to one type of codon, right? So this tRNA only has one anticodon, and this anticodon will correspond to only one codon in the actual mRNA strand. So then it will connect with this codon. Now, after it complements with this codon, it reads these nitrogenous bases and says, this particular portion, this codon is a start codon. So I understand that I'm going to be starting my mRNA translation here. And then this tRNA leaves, but before leaving, it creates an amino acid. Now, I don't specifically remember if start codons create an amino acid or not, but let's just say for this example, it does. Please talk with Ms. Soares if they do or not. I'm too lazy to search it up. Anyways, it creates an amino acid on the other side. Now, anticodon is at the bottom end of the tRNA, while the amino acid is at the top. So it decodes and realizes, okay, this is the codon connecting, and this also creates a type of protein. Which protein? The start protein. So I'm gonna start amino acid. So I'm gonna push the start amino acid here, and then what happens next is it continues on. It's done with coding this codon. Now it's gonna move to the next codon, but this anticodon doesn't correspond with this codon right here. So this tRNA actually leaves, but goes away and leaves the ribosome, but the amino acids that connects to the tRNA also releases from their tRNA and just stays there. Then the next corresponding tRNA with the anticodon that corresponds to the next codon in the mRNA strand comes in, they connect, and the next mRNA is next amino acid is made. And after this, these two amino acids connect. And this continues on and on and on and on until the entire mRNA strand is done. And after that, it creates a polypeptide chain, which looks something like this. And then at the end, it will then create a protein. So that's the whole concept of transcription and translation. I'm trying to minimize this video a lot because I remember that last time I took a lot of time explaining my stuff. I hope you guys understand. If you have any questions, talk with me tomorrow for it. I think we have a quiz on Monday. I don't know if we do. But if you need any help doing homework or anything, I will definitely explain. Thank you guys so much and have a